Okay, and welcome back to video 2 of echolocation in bats, where we're going to move into the physiology side of things, the neurophysiology, and talk first about sensory transduction of sound and the sensory receptors for sound in mammals. And this is common across all mammals, which is signal transduction by hair cells. And then we're going to end with a discussion about a tonotopic map that's created by the cochlea and then preserved up into the brain by labeled lines. And if none of those words make sense to you, that's okay. We're going to talk about them. Okay. So first of all, just in general, for sensory transduction, our sensory systems utilize specialized cells. And what those cells are supposed to do is to transform the energy of a stimulus, whatever that energy might be, into an electrical signal that the nervous system can understand. So they're translators that communicate information about the internal or external world to the brain and to the nervous system. So for different kinds of sensations, the energy that comes from the stimulus might be different. So for visual systems, the energy is photons, whereas, or it's electromagnetic, in other words. And in sound, in the auditory system, it's physical movement, because sound is physical movement of air, and the ear is good at detecting that. So we're going to talk about two functional roles of sensory receptors. The first is that they need to transduce the energy form of the stimulus into the electrical signal, or in other words, a receptor potential. And then second, they need to encode information about that stimulus. And in the bat system, we'll talk a lot about higher level computations in the brain for doing that. So we'll start with transduction of energy form from sound waves into action potentials. So ears translate mechanical movements, mechanical energy into electrical signals. And sound is a series of pressure waves, okay? And they have an amplitude or a loudness, and they have a frequency or a pitch. So here's some examples of this. If you have low amplitude, which is the height of the wave, tall versus short, then you have a quiet sound. So this height of this wave is short, so it's a soft sound. This height or amplitude is large, so it's a loud sound. And the distance between the waves is the frequency. So this is a low, soft sound. This is a low-pitched, loud sound, whereas this is a high-pitched, soft sound and a high-pitched, loud sound. So the sound waves, what our ears do is they make use of the fact that sound waves can cause physical vibrations of a membrane called the tympanum or tympanic membrane. And this physical movement is eventually detected by mechanoreceptors that can actually measure the physical movement of fluid in the ear. Auditory organs utilize the mechanical energy in sound in order to create action potentials. And so this means that they have to be physically stimulated, the receptors. And in bats and in mammals, this is the cochlea. And what the cochlea is it's your inner ear, and it's a long rolled up tympanum that's a variable thickness, and it vibrates with fluid movement inside the ear. And mechanoreceptors, and they're called hair cells, that are embedded into that membrane can detect the movement. So we need to convert mechanical energy or sound waves into electrical energy. And I just have a reminder here that a sensory neuron requires that an excitatory ion channel allows enough sodium into the cell to reach threshold for an action potential. And this typically occurs in response to excitatory neurotransmitters released from either the presynaptic neuron, which is what we've kind of been talking about in the background literature, or a sensory receptor, which is what we're going to talk about now. So the primary auditory neuron is listening to the hair cell, and it's listening to neurotransmitters from the hair cell. So this is a hair cell, the light orange thing here, and the auditory cells, these hair cells, release excitatory neurotransmitters when they're physically moved. So these weird structures at the top that looks like hairs, this is what they're named after. Those are actin filaments, and they actually extend out so that the light pink kind of embedded part around the cell body of the hair cell is the membrane. And then these hair cells, the actin filaments, stick out into the fluid surrounding the basilar membrane, and that's what's going to physically move. So the little hair cells on the top up here, these are going to wave back and forth with the fluid movement. And then if the cell is depolarized enough, then voltage-gated calcium channels open and allow calcium to come in and cause exocytosis and of neurotransmitter. And this is an excitatory neurotransmitter, it's glutamate, and it's going to tell the afferent primary neuron to fire an action potential. And that's how we have now translated physical movement up in this fluid, which is coming from the physical movement of the ear bones and the membrane, the fluid inside of the cochlea, into a chemical and finally an electrical signal that neurons can make sense of. So here's what they look like in a micrograph. So you can see this is the surface of the basilar membrane, and here's the actin filaments sticking up out into the fluid. And these little actin filaments are going to wave with the movement of the fluid in the cochlea. And then these blue arrows are pointing to what are called tip links between the actin filaments. And these are super important because this is actually how the physical movement is going to open and close channels that allow ions to depolarize the hair cell itself. So in this case, you don't have neurotransmitters or something depolarizing the hair cell, you have physical movement. So what happens is as the actin filaments are pushed in one direction, this connection, this green tip link here, actually pulls open a potassium channel, and potassium floods the cell. And it's a little bit unique in that it's potassium from other neurons, which usually use sodium to depolarize. I'll tell you why that, or we'll talk about why that is in a minute. But so you have these little stereocilia, these actin filaments that are waving in the fluid, and every time that they move, they're depolarizing the cell. So the similarities to neurons are that a depolarization event causes voltage-gated calcium channels to open, and the calcium presence causes the vesicles to fuse and exocytose glutamate onto the primary sensory neuron that's waiting there and listening to it. What's unique about hair cells is that I just mentioned they use potassium to depolarize. And I don't know why that is, what the evolutionary kind of history of this is, but it turns out that the fluid that's inside the cochlea that is bathing these stereocilia is really high in potassium. And so when these tip links are pulled open, potassium floods the cell and depolarizes it. And it goes in because of this very high concentration outside of the cell. So you're driving potassium concentration up inside the hair cell. That opens the voltage-gated calcium channels, which then allows vesicles to fuse into the synaptic cleft and excite the primary sensory neuron that's listening to it. It's easy to get potassium back out of the cell. You don't have to, in fact, the cell doesn't have to use energy to do it, because just like any other normal extracellular fluid, the extracellular fluid inside the basilar membrane, this extracellular space here, is low in potassium, so it can just come on out. The body is going to have to use ATP in order to pump that potassium back into the fluid in the cochlea, the cochlear fluid. So that's where you pay for it, right? You pay the bill on the back end, creating this high potassium fluid in the ear. Okay, so potassium is depolarizing the hair cell rather than sodium. The cell body is surrounded by normal extracellular fluid, and so that the potassium can leave the hair cell again. And so you get pretty fast sodium coming in and then sodium going out. And so what you see is this kind of cyclical signaling, because when the tip links are pushed one way, it opens the potassium channels. They're physically pulled open. It's literally like opening a gate, you know, that you pull. The tip links move and it pulls open the potassium gates, and potassium floods the cell, calcium floods the cell, and you get transmitter release. When the tip links get pushed in the other direction, so this is extremely fast, right? The potassium channels close, and the potassium is so rapidly leaving the cell that you get a hyperpolarization event very briefly. So you get this pulsatile release of neurotransmitter, where in one direction you get a bup, 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 bup firing of the action potential of the auditory nerve. So this is an action potential in the auditory nerve that's listening to that hair cell. So it's firing, 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 firing. It's getting excitatory glutamate dumped on it a bunch of times. And then when the hair cells move in the opposite direction, then the stereocilia move in the opposite direction, and the tip links close, you get a silence event here. And so you get this cyclical wave that tells the neuron that that section of the basilar membrane is vibrating. I put it that way specifically because the hair cell can only tell the auditory neuron that it's moving. It is being physically moved. But there's nothing about the hair cell's signal that encodes frequency information. So we're going to talk about that next. Like, what is it about how can you get that information? How can you tell if this is a high pitch sound or low pitch sound? And what we're going to see is the cochlea has a tonotopic map on it, or creates, sorry, that's not right. It creates a tonotopic map based on its morphology, and we'll learn about that soon. And then labeled lines come out of the cochlea, and they're highly organized in how they attach and connect to the structures down the line, all the way up to the auditory cortex. So first, let's start with the cochlea. So we need to know what frequencies are in the sound. You guys are able to understand my language right now because you've been taught what the frequency modulation of saying the different letters and words mean. But part of that means that you have to be able to even detect and differentiate that there are different frequencies in the sounds that I'm making. So what happens is the pinna catches the sound, which gets funneled down into the ear, and the eardrum, the malleus, the incus, and the stapes their job is to basically pound out the frequency wave onto the round or the oval window that leads into the cochlea. And the cochlea looks like this kind of little snail thing inside the ear. It's basically a long structure that has been rolled up to save space. So that's why it looks like that. There's also semicircular canals here, which is how the bat understands where it is in space, if it's this way or that way or upside down or whatever. We use the same system for us. So here's the cochlea unrolled. And so you get what's called a tonotopic map, just like you have on a keyboard. So I put this here as a reference for what a tonotopic map is. So if anybody has taken your hand and run your fingers up and down a keyboard, it goes from low frequency to high frequency and then back down. That's a tonotopic map. And you can play music of very complex sounds and frequency combinations, in other words, chords, because it's mapped out, and a musician trains themselves to know which keys to push at the same time, right, to make those sounds. The basilar membrane also has a tonotopic map, but it's created by physical properties. So the basilar membrane is this light pink part underneath the cochlear duct, which is where the fluid is. So that's where the hair cells are embedded in the basilar membrane, and they're sticking their stereocilia out into the cochlear duct, which is where the fluid is. So this membrane extends the whole length of the cochlea, which is then rolled up into this shape inside the ear. But the basilar membrane is not a uniform thickness throughout. It actually changes. And near the start of the cochlea, so right at the oval window, sorry, the round window, that was the oval one, anyway, whatever. The bones are beating out the frequency right here at the oval window, and that is then, that wave motion is then transferred from the air to the fluid in the cochlea that bathes this basilar membrane. And a cool thing about the basilar membrane is that it's going to vibrate differentially depending on its thickness. So you have a thick section at the base here, and then you have a thin section at the other end down here. And it takes a lot less energy to make a thin, say, piece of paper wave than it is to make a piece of cardboard wave. So what you essentially get is that if you have a high frequency sound, it will excite and move the hair cells that are at this thicker part. If you have a low frequency sound, it can't move this thick part. But as you move down, there's going to be some point where the hair cells start to vibrate most excitedly. And that is going to be dependent on the frequency matching the thickness of the membrane that it best vibrates. So you get what's called a tonotopic map from high frequency to low frequency. That's purely generated by physics and the thickness of the basilar membrane that these hair cells are embedded in. So some points: the actual hair cells that are embedded in this whole membrane are all identical. What makes them release neurotransmitters in a pattern that's specific to the membrane or to the frequency is where they are in the membrane and the thickness of the membrane. So you end up with a tonotopic map of high frequency at the base, and then low frequencies at the apex, the tip. So that's cool. You have now a way to create a map of frequencies in the basilar membrane. But now you need that information to be transferred up to the brain in a way that's organized, so you don't lose that spatial information that now means frequency. So that is achieved through connectivity, moving from the cochlea and that tonotopic map all the way up to the auditory cortex. So we call these labeled lines basically when there is kind of conservation of spatial organization. And one thing that's cool about labeled lines is that the perception of a given sensation is dependent on the receptor type that transmitted the information on the pathway up to the cortex, not on the type of stimulus that actually causes the receptor to fire. And the only reason I'm saying this is because there's kind of cool examples where you have accidents, essentially, where something that's not meant to turn on a sensory receptor turns it on, and you perceive that sensation as if the sensory stimulus was there. So I'm going to give you two examples. One is on this slide. So you can perceive or see spots because photons excite your photoreceptors, which is the normal case, or because you push on your eye and it actually stimulates the photoreceptors to fire, or because you hit the back of your head and you actually stimulate the visual cortex to fire. completely independently of the receptors themselves. In all of those cases, you'll perceive flashes of light, but in the first case was the only time that there actually were flashes of light. That's kind of this labeled line phenomenon. Here's another cool example. Menthol, that's in mint flavors, actually activates cool temperature receptors in your mouth. So mint is not actually physically cold in temperature, but it's perceived by you as cold because it activates temperature receptors, not for any other reason. And the opposite of that is capsaicin, which activates heat receptors. So we all know that you can touch a pepper, a hot pepper, and it's not actually physically hot, but it is perceived as hot and truly perceived as heat because it activates hot, warm temperature receptors. So this is an example of a labeled line. You're perceiving something because the neuron that's the sensory receptor that's activated through whatever means is telling your brain, when I fire, I mean it's hot. When I fire, I mean it's cold. And so in sound, when a hair cell fires, your brain perceives it as a particular frequency. And that's important. That's how we can make sense of sounds. So receptors are sending information along a neural network, which means a connection of neurons. And the connection of neurons is going to be very ordered so that when that information reaches a destination, in this case, in this picture, a cochlear nucleus, but it could be all the way up to the auditory cortex in the brain, where you actually have a perception that something happened. That spatial relationship is super important to keep organized. And so we're going to call this spatial preservation. And this allows the cortex, all the way up in the brain, to interpret information as a particular frequency. So if we look at this picture, you have the base of the basal membrane, which is thicker. The hair cells here, if they fire, they're going to transfer their information along this green neural network. Okay, so these might be multiple neurons that are finally synapsing on a destination. And essentially what I want you to see is that the physical relationship between these three hair cells here, this one being the highest frequency hair cell, and then moving down to the lower frequency at the apex, is conserved so that the lower frequency is still at one end of this structure, and you move up in frequency as you move towards the other end of that structure. So there is spatial preservation of the tonotopic map in the cochlea to a tonotopic map in the next structure. And that's going to happen all the way up the line. So here's an example of a bat basilar membrane where you have of the acoustic fovea, which I've highlighted in red. So one thing about a bat's basilar membrane that's kind of cool is that it'll be kind of normal shaped, a normal mammal width of the membrane, across a range of kind of the broader frequencies that say you and I hear. But then when you get to its specific echolocating frequencies, these high frequencies, the basilar membrane actually widens out and gets bigger. So there's more hair cells dedicated to those frequencies because those frequencies really matter. So in this species, this example, the CF component of its call, the frequency that it uses in its echolocation is 83 kilohertz. And so you can see that while this whole half was dedicated to 60 kilohertz, the rest of this is dedicated to about 10 kilohertz. And so there's a much bigger area in the cochlea dedicated to that frequency. And that, again, because of spatial preservation, also means that there's a bigger area of the cochlear nucleus that is dedicated to those frequencies. And then the cochlear nucleus is going to send that information down the line all the way up to the auditory cortex. And there's more of the auditory cortex that's dedicated to this frequency range. So you have, this is what the acoustic fovea is. More hair cells that are tuned specifically and probably tuned to very small differences in this kilohertz range that's really important to the bat's echolocating. So here's another just visual representation of this tonotopic spatial preservation. So we're starting down here in the cochlea. You see the rolled up structure, and it's color coded by frequency. So low frequencies are blue, high frequencies are red. And so the cochlea is creating the tonotopic map, like we talked about with the thickness of the basilar membrane, and then spatially preserving its connections up to the cochlear nucleus, CN, right here. And then you can see there's a whole bunch of structures that in every single one, there is some kind of spatial preservation of the information so that it's ordered and that there's information about frequency based on the location of the cells. And that connectivity goes all the way up to the auditory cortex in the brain. This is going to be kind of where the highest level of processing and understanding and perception occur. All of these other structures in between are modifying information to make things more precise and more informative. And in some cases, even making calculations about distance or speed, so that detecting the Doppler effect, and sending that information up to the auditory cortex, where the bat can finally understand that's a moth I can eat, and it's 50 feet away, flying up, and it's going to assess that information the whole approach up to that bat. Okay, another point about this, you see ascending information in red, descending information in blue. All that means is that basically there's a whole lot of neurons that are sending information in both directions between these structures. So this picture is helpful in that it demonstrates the tonotopic spatial preservation, but also in that you can see how complex the connections are between these structures, and therefore how complex it is to process this auditory information and create a picture of the world for the bat. Okay, so I'm going to pause there and we're going to go into video 3, where we're going to talk more about the specializations and the actual calculations in this processing pathway.